Hello everyone, today we are going to learn about RNA interference or RNAi. Before starting, let's dig into the history of RNAi. Do you recognize this flower? Yes, it's petunia. You may have this in your garden. If we put an extra copy of the gene for a pigment-enhancing enzyme to this petunia, what will be the result? Flower with darker pigment, or the lower one with variegated petals with sections of no pigment? Most of you will expect the upper one. Scientists who were doing this experiment expected the same thing, but surprisingly they got the lower one with less pigment. But how could adding extra copies of gene for enhancing pigment result in less pigment? The researchers measured the mRNA level of the pigment-enhancing enzyme and found it significantly reduced. It is now clear that reduced mRNA resulted in less pigmented flower. Now the question arises, how could introducing extra copies of the gene reduce the amount of mRNA? The answer actually came in the year 1998 from researchers working on the nematode Caenorhabditis elegans. It was observed that when double-stranded RNA was injected into the worm, it led to a decrease in the expression of the homologous gene. This results into a movement defect, twitching behavior. These studies indicated that double-stranded RNA seemed to be critical for gene silencing. Gene silencing mediated by double-stranded RNA has been named as RNA interference or RNAi. With clues from the C. elegans research, scientists found that plants have a special RNA-dependent RNA polymerase that produces double-stranded RNA molecule when triggered by a very high mRNA level. In response to the high level of mRNA, this RDRP creates the double-stranded RNA molecules which shut down the pigment gene expression in petunia. Andrew Fire and Craig Simello were awarded Nobel Prize in Physiology or Medicine for their discoveries related to RNA interference. RNA interference is an evolutionary highly conserved process of post-transcriptional gene silencing by which double-stranded RNA causes sequence-specific degradation of mRNA sequences. The RNA molecules those function for silencing genes are short, usually about 20 base pairs long. The basic macromolecules involved in the RNAi mechanism are two types of RNA molecules, microRNA or miRNA and small interfering RNA or siRNA. These two types of RNA can bind to a specific mRNA through complementary bonding and inhibit the translation by ribosomes. The miRNAs are short, non-coding RNA nucleotides produced from the chromosome inside the nucleus and have hairpin sequences folding them into double strands. On the other hand, siRNAs are exogenous dsRNA which are similar to that miRNA but not produced inside the cell's nucleus. First, coming to the gene silencing mechanism of siRNA. An enzyme named dsR recognizes and cuts the double-stranded RNA into short 21 to 25 base pairs long molecules called siRNA. The siRNA interacts with and activates the RNA-induced silencing complex or RISC. The endonuclease Argonaute 2 component of RISC cleaves the passenger strand or sense strand of the siRNA. In contrast, the guide strand or antisense strand remains associated with the RISC. The guide strand guides the active RISC to its target mRNA. It only binds to mRNA that is fully complementary to it. Now the AGO2 cleaves the target mRNA and causes specific gene silencing. Similarly to siRNAs, miRNAs also inhibit gene expression in a post-transcriptional manner. miRNA gene transcription is carried out by RNA polymerase II in the nucleus to give primary miRNA or pri-miRNA. The pri-miRNA is then cleaved by a microprocessor complex comprising of Drosha and microprocessor complex subunit DCGR8 to form precursor miRNA or pre-miRNA. The pre-miRNA is subsequently transported by Exportin 5 from the nucleus to the cytoplasm. In cytoplasm, it is further processed by dicer into a miRNA duplex of 18 to 25 nucleotides. The miRNA duplex then associated with the RISC forming a complex called miRISC. The miRNA duplex then unwinds, releases and discards the passenger strand or sense strand. The mature single-stranded miRNA guides the miRISC to the target mRNAs. The miRNA binds to the target mRNAs through partial complementary base pairing. The target gene silencing occurs via translational repression, degradation, and cleavage.